Hello everyone. I'm very pleased to share with you today our team's recent work published in iMetaOmics, titled Diversity, Transfer Potential, and Transcriptional Activity of Virus-Carried Antibiotic Resistance Genes in Global Estuaries. In recent years, intensive human activities have discharged large amounts of antibiotics into estuarine environments, driving the enrichment and spread of antibiotic resistance genes, ARGs, posing serious threats to the health of coastal residents. However, our scientific understanding remains skewed heavily toward prokaryote ARGs. In addition to prokaryotes, viruses also act as significant reservoirs of ARGs. And they can also facilitate the dissemination of ARGs among prokaryotic hosts through viral transduction. Therefore, studying viral ARGs is key to assessing ARG transmission risk in estuaries. To comprehensively explore ARGs carried by viruses, vARGs, in estuaries, this study focuses on major estuaries worldwide. Specifically, we collected 60 samples from the Pearl River estuary and downloaded 624 datasets from public databases such as NCBI. Using these global estuarine metagenomic data, we aims to answer two key scientific issues: how vARGs contribute to the estuarine antibiotic resistance in terms of diversity, abundance, transmission, and expression. What factors influence these vARG characteristics? Using metagenomic technology, we constructed a first global estuarine virome dataset, and found that estuarine viromes are novel and diverse, and the diversity of estuarine viromes remains largely unexplored. Through stringent functional annotation, we found that estuarine viruses carried diverse vARGs. These vARGs were also widely distributed across estuaries. Estuarine viruses are not only reservoirs of ARGs, but also important disseminators. Viruses carrying ARGs have a broad potential host range, and those with higher vARG density tended to interact with more prokaryotic taxa. Based on minimal evolutionary distance analysis, we further found that viruses mediate diverse horizontal ARG transfer events. Viral ARGs are not merely transferred passively, they can also be actively expressed within host cells. Metatranscriptomic data analysis revealed that 51% of vARGs are actively expressed in estuarine environments, with some even showing higher relative expression levels than their prokaryotic counterparts in the same samples. To explore the environmental drivers affecting vARG characteristics, we performed correlation analysis, random forest analysis, and structural equation modeling. We found that temperature and human activities can directly or indirectly increase the environmental risks of vARGs in estuarine water. Although the mechanisms that underlie these phenomena are not clear, we proposed some possible explanations. On the one hand, higher temperatures could increase prokaryotic metabolism and reproduction in estuaries, and in turn, boost viral replication and infection. This increased viral activity may subsequently facilitate the proliferation, transmission, and expression of vARGs. On the other hand, increased antibiotic use result in more antibiotic residues entering into the tropical/subtropical estuaries, which may increase the selective advantage of ARG-carrying microorganisms and consequently facilitate the spread of vARGs. Overall, our study presented the landscape of vARGs in global estuaries and highlighted their potential risks, suggesting the necessity of incorporating vARGs into future surveillance and management of estuaries, especially in the context of climate warming and social development.